I'm Xuan Qu. I'm class of 2017. I'm a neuroscience and math major, and my research aims to understand the stretch feedback pathway in the cardiac ganglion of the American lobster. Dickinson lab looks at neuromodulation on the central pattern generator, and central pattern generator are generally neuronal networks that controls rhythmic movements, for example, in our body, walking, swimming, and swallowing. And so one question is how we produce a very flexible output of our movements. For example, when you walk around, there's different modulation on the pattern generator to give you flexibility in your behavior. My project looks at both the intrinsic neuromodulation and extrinsic neuromodulation on the central pattern generator, the cardiac ganglion, in the lobster heart. My hypothesis is that there are mechanosensitive ion channels present on the stretch-sensitive dendrites in the lobster heart, and they transduce the stretch information back to the cardiac ganglion, forming a stretch feedback loop. And we're still trying to identify the specific ion channel that are involved. So in my experiment, I isolate the cardiac ganglion along with muscles surrounding and underlying the pacemaker cells, and I do a tonic stretch on the muscle. This aims to mimic the heart contraction in lobster. And in the response, we normally see a phase delay during increasing stretch and increasing burst frequency during hold period and increasing burst duration during return from stretch. By looking at these changes and trying to understand why they happen, we can have more ideas of what type of ion channels that might get involved. For example, the phase delay, it might be a hyperpolarizing current, so it could be a cation moving out of the cell. And for the increasing burst frequency, it might be a depolarizing current, so it might act through another ion channel, have cations moving into the cell. And so the future direction will be we've built separate transcriptome for the premotor and motor neurons, and we're trying to identify the potential mechanosensitive ion channels via homology, ion channels that have already been identified in other species. And also another approach will be through mass modeling. We basically build a model for the cardiac ganglion, and we have different expressions for different possible ion channels, and we just try out different combinations and look for the closest response of the computational model with the biological system.